So next, almost the last topic of genetics is translation. So translation means to convert the language into a meaningful in the language world. We have translation means to convert one language into another. So basically translation means to make the things in a meaningful manner. So translation in genetics, translation mean actually the to make the end product of the gene, that is the protein. And we have known that the genetic transcription, or the initial transcript, or the inscription of the uh, genetic into a genetic language is known as transcription. And in the transcription, it synthesize into 5 to 3 direction. DNA replication happen into 5 to 3 direction. And in the translation, we have known that this synthesis is from protein synthesis is in the N terminus to the C terminus. So the translation happens from N terminus to C terminus. And for translation, we have three important processes. And the first process is initiation. For initiation, obviously we need an initiation factor. So for initiation, we need eukaryotic initiation factor, which does identify, which helps in assembling of a thing, and which then releases post assembling. So it identify the transcripted part of the newly formed messenger RNA 5' gap, and bind that messenger RNA with a ribosomal 40S ribosomal unit. And at this ribosomal unit, at the P site, it bind an initiator, methionine initiator charged transfer RNA. So elongation factor is actually helps in translation by initiation factor helps in translation by initializing this translation. And this initializing is important, and it is mediated by initiation factor, which identify that 5' and after identification, it helps the assembly of 40S ribosome and a charge transfer RNA having methionine as a start codon on it, and bind them on the P site and on the messenger RNA, and then releases. So it has three important functions. So this initiation process, it requires GTP, and for G we need to grip things. We need to grip that 40S ribosome and the initiator transfer RNA to the messenger messenger RNA. So after initiation, now the process elongation is in the translation. The elongation happens when elongation is also GTP mediated. Why? Because we have known that we need to grip things. We need to grip methionine. We need to grip other amino acyl transfer RNA which are charged with ATP and amino acyl transfer RNA to grip or to bind the messenger RNA at the site of A. So A for amino acyl site. So after methionine initiate the translation, now the at the A site the charged transfer RNA which are scattered in the cytoplasm, they come and now one by one they attach to the A site of the ribosomal subunit. After amino acyl come and grip that messenger RNA site at the A site, now a ribozyme come or ribosomal RNA, rRNA, ribozyme come which catalyze and transfers. Catalyze and transfer what? The growing chain where? To the P site or the polypeptide site. Growing chain mean the polypeptide chain. So after binding of the initiator methionine, initiation starts. Now we elongate like one code, second code, third code, fourth code. So code by code the messenger RNA detect that code, ribosomal RNA move, and then and irrespective to that code the charge transfer RNA taking the amino acid with them, they come and they give that amino acid at the A site. And when they come at the A site, now two transfer RNAs are present at the A site. So this transfer RNA, it gives the which are incoming at the A site, it now gives its amino acid to the P site, which is the growing amino acid chain. It gives to that amino acid and then releases. So that P site or the polypeptide site is growing all the way up. So the next step in the elongation process, now the second transfer RNA gave that amino acid to the first A site transfer RNA, and the chain is elongated. Now it elongated, go and on. So after giving that after catalyzation, so now the ribosome, not the messenger RNA but the ribosome, it get translocate or it moves 3 nucleotide or 1 codon towards the 3 end on the messenger RNA. And this process need GTP. This translocation need GTP. So now we have one GTP used by the initiator, one GTP used by the coming transfer RNA, then the next GTP is used by translocation process. So after translocation, now the chain is growing on and the ribosome translocate further, and after this the termination happens by there is a release factor, eukaryotic release factor. But before going to that release factor, we have known that diphtheria and pseudomonas they inhibit this elongation factor, which we will discuss here. Which make the chain to elongate. So diphtheria toxin actually inhibit that elongation factor and inhibit the protein synthesis and cause diphtheria related pseudomembranous corynebacteria diphtheria and Pseudomonas aeruginosa. So it's an important link to microbiology. So going to the termination, we have the elongation release factor, and this release factor, it recognizes a stop codon. We have known that the stop codons are you go away, now you are away, now you are gone. The release factor recognize them and terminate or halt translation. So we need GTP in order to release this growing chain transfer RNA or the growing chain. So the growing chain detach itself from the transfer RNA, and the transfer RNA need GTP energy to release off. So this is whole mechanism of translation. So what happened to that translated protein? So post translation modifications include, we can trim it, we can trim or we can remove the propeptide things, like in order to use trypsin, we have a pre-pro type of peptide trypsinogen, and in order to get activate that trypsin, we need to remove the N-terminus propeptide, its N-terminal, and hence make it functional. So trimming is post-translation modification, and also we have a covalent modification. We can phosphorylate the chain, we can methylate the chain, we can acetyl the chain, we can also we can send it to ubiquitisome or ubiquitination, or also we can make them glycosylate, we can make them hydroxylate, and we can use it in different pathways. So this is the post transcriptional modification. And after this we have one thing in prokaryotes. We have a sequence known as the Shine-Dalgarno sequence, which actually makes the ribosome, which actually the ribosomal site on the prokaryotic messenger RNA. It enables translation by just aligning the ribosome to the start codon, and it gets started translation. Regarding genetic code information, the genetic code is always universal, means it is conserved throughout the species, throughout the evolution, except the mitochondria and some bacteria and other species, they deviate from their universal genetic code. The next thing is the genetic code is commaless or non-overlapping. What does it mean? That we can read it from a specific start point in a continuous manner without any interruption. And it is commaless, but there is exception that some viruses have a different type of genetic code, except some viruses. Then we have unambiguity or unambiguous, and for unambiguousness means that there is a clear-cut thing. One codon can make one amino acid only. The next thing is it is degenerative and redundant, and it means that 20 amino acid is produced by multiple or 60 codons. 60 codons can make 20 amino acids. Then we have the Wobble hypothesis, which tells us that only first first two nucleotides are required for traditional base pairing sequence. The third one, it can be differ, but it is not required for traditional base pairing, and it can code for same amino acid transfer RNA pair. So this is all about the genetics, and now we are at the verge of ending of this genetics section.